investigator at the Scintillon Institute in San Diego, California. If you've been in the field of extracellular vesicles for any time, you probably are familiar with John and his work. John has been a key person in the area of flow cytometry and extracellular vesicles. Part of the EV flow cytometry working group, which constitutes a number of interested investigators studying EVs using flow cytometry as a evaluation tool. And as part of this EV flow cytometry working group, a recent paper has been published as a position paper in the Journal of Extracellular Vesicles, which is the topic of our discussion today. And that topic is the framework for standardizing the reporting of extracellular vesicle flow cytometry experiments, otherwise known as MIFlowCyt-EV. So John, thank you very much for lending your time today and sharing this information with the audience. And a reminder, please put all of your questions into the Slack questions channel, the EV seminars questions channel.
Thank you, Joanne. Welcome, everyone. Thanks for coming. So there's a title of my talk. I'm representing the work of a number of people who I'll introduce in a minute. As Joanne mentioned, a group of us have been working on figuring out how to talk to each other about our EV flow cytometry experiments, and recent manuscript that summarizes some of that work is going to be our topic today. So there's the title. There's a supplementary title, Flow Cytometry EVs, What Could Possibly Go Wrong? And the things I'm going to tell you about today are the result of hard lessons many of us have learned trying to use flow cytometry to measure vesicles. And so this is the first of a series of educational talks on different topics. I'm going to give a broad overview here today, but we'll be having more presentations and discussions about individual topics in the future. So here's a summary of what I want to cover today. I just want to describe, what are we trying to do when we use flow cytometry to measure vesicles? What are some of the challenges for using flow cytometry to measure EVs? And then I'll go step by step through the reporting guidelines and describe rationale behind some of those guidelines and give some examples for some of the more important topics. So as Joanne mentioned, I have a lab at the Scintillon Institute in San Diego. We develop technologies, flow cytometry mostly, instrumentation, reagents, assays. We do a lot of our biology by collaboration, including with the ERCC group, Extracellular RNA Communication Consortium from NIH. We work with a consortium of pharma scientists working on biomarkers and EV FC working group. We have some commercial activities as well. I won't talk about those, but you can Google those and learn about those at your leisure. So what do we want to measure when we try to use flow cytometry to measure vesicles? It's pretty simple, really. We want to know how many there are, the number of vesicles, or more often the concentration. So we need to count them. We need to know how much volume we measured. We want to know something about the size, because size is a distinguishing feature of EVs. But probably the most important and exciting area of EV research is understanding the cargo vesicles carry, both on their surface and inside of them. And this is where flow cytometry has great potential. So, in general, we can boil our objectives for flow cytometry of EVs down to these three topics. Presenting these guidelines, against the backdrop of other initiatives to promote reproducibility and rigor in science. In the area of vesicles, it's become apparent that there are some issues with rigor and reproducibility. And an interesting read is the EV track work from a European group that's basically been cataloging the methods used to process and analyze vesicles. And there's a lot of interesting insights in this paper, as well as some lessons. As most of you probably know, the most common approach to fractionate biofluids to enrich or isolate vesicles is ultracentrifugation. A pretty simple looking 3-step centrifugation protocol that you'll find in, turns out, about 50% of the papers. Typically, there's a low speed spin, a higher speed spin, and then an ultracentrifuge speed spin to make some different fractions. The EV track group cataloged the reporting of those methods. They found that there's actually more than 200 ways to perform that spin. Those spins, and probably a lot of the reproducibility issues in the literature can be traced to just those differences alone. Whereas flow cytometry is a lot more complicated than centrifugation, so there's even more ways to go wrong. ISEV is addressing the broader issues of reproducibility by laying out guidelines for should report. Most of these focus on conventional biochemical analysis, Western blots, ELISAs, etc. A few mentions of single vesicle analysis, but it's an emerging area, so hard to be definitive about recommendations there. So it's a gap that we're looking to fill. In terms of flow cytometry of EVs, most of the challenges can get boiled down to 3 main topics, which are summarized here and addressed in the manuscript. The first has to do with misunderstanding about how to use light scatter, interpret light scatter measurements. We all learned in kindergarten that we use light scatter to detect cells in flow cytometry, and that forward scatter is a measure of particle size, and side scatter is a measurement of cellular granularity. Those are convenient, simplifying concepts, but they're both wrong, and they're especially wrong when it comes to vesicles. So light scatter from vesicles is actually very complicated. And so I'll make some comments about how to properly interpret light scatter as we go here. The second big pitfall in flow cytometry of small particles is coincidence. And this is the case where a single particle is too dim to detect. But if you fit enough of them into the probe volume of the instrument, the laser beam, you can measure something. But it won't be a single vesicle, it'll be many vesicles. And so this is a coincident artifact, also referred to as swarm, is a pervasive artifact in the literature. But there are ways that you can evaluate its presence and avoid it. The third big area is fluorescence calibration, or rather lack thereof. So without having calibrated fluorescence axes, it's really hard to interpret data, especially when your particles are very dim and near the background noise of your instrument or assay. So a lot of the data that's been published is really hard to interpret because it lacks. And these are issues that are addressed in the guidelines, and I'll touch on those. So, you know, these pitfalls or challenges, you know, were recognized by a number of people independently as they tried to measure vesicles by flow cytometry, and the whole set of papers that have described the various artifacts that can appear when you try to measure dim particles by flow. And so this is where the field was, say, 5 years ago. A group of us got together to compare notes. People have been trying to measure vesicles by flow cytometry for several decades. About 10 years ago, a group within ISAC, the International Society for Advancement of Cytometry, started having workshops and small meetings to discuss this. About the same time, ISEV was formed, the International Society for Extracellular Vesicles, and a lot of us started running into each other at these meetings. And you know, the initial goal was to figure out the best way to measure vesicles. After a few rounds of discussion, we realized that we weren't even talking the same language, that everyone had a different instrument, everyone was using their instrument in a different way. People were reporting their results in different ways, and we realized we had to take a step back and work on how we report what we did and what we measured. So that was a couple-year process of discussions and workshops. If you attended the CITO meeting or the ISEV meeting or the ISTH meeting, you may have gotten to sit in on some of those. And the end result is a manuscript that was just published in the past couple of months. It's intended to complement these existing guidelines documents. So there's already guidelines for how to report on vesicle experiments. That's the MISEV guidelines. There are guidelines for how to report on flow cytometry experiments. Those are the MIFlowCyt guidelines. And these guidelines are intended to sort of fill the gap between those two sets of guidelines. And so if you read the manuscript, which I recommend everyone does, you'll find we broke the process down logically from sample preparation to data reporting and sharing into these different categories. And you know, when we talk about flow cytometry of vesicles, a lot of discussion is about the performance features of different flow cytometers. The most frequent question I get is, what flow cytometer should I buy to measure vesicles? And that's a hard question to answer because measuring vesicles depends on a lot more than just the instrument. The instrument is important, but the instrument is only part of an assay. So all the things that you do to a sample before you present it to the instrument, as well as how you use the instrument, can affect the results. And so you'll notice that most of the elements of these guidelines refer to the assay independent of the instrument. And so it's I think useful to keep in mind that there's a difference between instrument and the assays that one would run on the instrument. So I'm just going to step through each of these 7 areas one by one, describe briefly what the rationale behind them is. As I mentioned, we'll have future presentations, and there's lots of area for discussion on all of these sorts of things that will hopefully ensue. I'll give a couple of what were considered some of the most important examples that were highlighted in the manuscript, but really all of these things are important. You have to do a lot of things right in order to make a interpretable measurement of vesicles by flow. So step 1, category 1, pre-analytical variables and experimental designs. These have to do with Where'd you get your sample from? What did you do to it before you prepared it for analysis? How did you set your flow cytometer up? What are the characteristics of your flow cytometer? These are all outlined in the existing documents, the MISEV and MIFlowCyt documents. And so those are, incorporated here by references. We're ask that everyone follow best practices for both EV and FC research. Category 2 is sample preparation. So these are really the way, the core of the assay. It's what you did to the sample, what you used to stain it with, concentrations of reagents, how long you stained, did you do any washing or other processing steps, and did you keep track of your dilutions and concentrations along the way? Can you all hear me? Joanne?
We can hear you.
Great. Because we want to learn the concentration. So if you did a concentration by ultracentrifugation or another method, recording the starting volumes and final volumes is necessary to actually calculate the concentration. Often these details are missing from publications, and it makes it really difficult to interpret or repeat experiments. Step 3 has to do with assay controls, and there's a whole set of assay controls that are listed here and described. And each of these controls actually is designed to address a specific artifact that you will encounter when you try to measure vesicles by flow cytometry. The first two, buffer only and buffer with reagents controls, are necessary to evaluate what sort of background might be in your sample just from those elements of the assay. It's the issue of specificity. How do you know that the events you detected are really vesicles? If we're going to do immunofluorescence, which is going to be our main tool for assessing cargo of vesicles, there's a whole set of controls that parallel what you would need to do for flow cytometry of cells. You want to be able to assess autofluorescence by measuring an unstained sample, because some receptors will bind to antibodies by their FC portion, that is, they are actually antibody receptors. The isotype control is a necessary control to evaluate whether you have this FC receptor type binding. If you're doing a multicolor experiment, it's important to have single stain controls, both to assess spillover of fluorescence from one channel to another, but also to decide where to set a threshold for determining whether something is quote unquote positive. There's a whole class of controls that are going to be assay dependent that we refer to here as procedural controls. So if you're doing a lot of processing to your sample, if you're doing several rounds of ultracentrifugation, or you're running things through a size exclusion chromatography column or other chromatography column, there's the opportunity for artifacts to be introduced here. So just as a general all-purpose control, if you have a involved sample preparation protocol, it's important to actually process buffer and reagents through that procedure, just to make sure that you haven't introduced some unexpected artifacts. I mentioned the coincidence artifact earlier. Nice way to evaluate the effect of that is to a serial dilution of samples, and I'll discuss that in just a little more in a second. And then finally, we expect vesicles to be sensitive to detergent. So detergent treatment is a useful control experiment to show that the particles or events you detected in your flow cytometer were actually due to vesicles because they were destroyed by detergent. So it's a lot of controls here. But as I mentioned, each of them, is designed to address a particular kind of artifact that can appear and confound interpretation. So for me, and I think for a lot of others in the group, hard to imagine being able to interpret an EV flow cytometry experiment without being able to see these control experiments. So they're really important for validating an assay. So here's a depiction of that swarm artifact. So on the left-hand side, we see a picture of a flow cell with some particles in it and a laser beam intersecting with the flow cell. You can see if the particle concentration is high, you might have many particles in the laser beam at the same time. And this is a purely statistical phenomena that depends on the concentration of particles in the sample. Most often when we're presented with a sample of EVs, we actually don't know what the concentration is. So a prudent initial experiment is just to do a dilution series to evaluate whether you have this coincidence artifact or not. And what you should find if you're truly measuring single particles is that as you dilute the sample, say in twofold steps, the event rate, particle count, should decrease in proportion to the dilution. But the brightness of the particles shouldn't change, because if you're truly measuring single particles, it shouldn't matter what the concentration is. The brightness of a particle is the brightness of the particle. On the other hand, if when you do this dilution series, the median brightness of your population does change, it's a good indication that you've probably got some coincidence going on. And so this is an important control to demonstrate that your assay isn't being confounded by this artifact. Okay, so those are all the controls that really should be part of experimental design. Now finally we're to the instrument. And there's a number of issues that need to be taken care of regarding the instrument and reporting how you use the instrument. And the first and most important is to specify what channel are you using to trigger detection, and what threshold did you use for that channel. I mentioned earlier for cell analysis, light scatter, forward angle light scatter, is most commonly used to detect vesicles. But that's probably not the best channel to detect vesicles with. Forward scatter works great for particles that are larger than the wavelength of light. Scatter in the forward direction is strongest for those particles. But as the particles get smaller, and particularly when they're smaller than the wavelength of light, that's no longer the case. Light scatter is more or less uniform in every direction, and side scatter may actually be a better channel to measure because it has lower background scatter. Whereas both light scatter is a non-specific signal, lots of things scatter light besides vesicles. Actually, vesicles don't scatter much light at all because they have a very low refractive index. And so a number of groups have pointed out and taken advantage of the advantages of fluorescence for triggering detection. And so whether you're using an antibody or a membrane probe or some sort of cytoplasmic probe, one can often get enhanced specificity vesicle detection. Fluorescence trigger. whatever you use, it's important to report it. And so, if you read the literature, particularly for a year or so ago, you'd find a lot of papers where it wasn't actually stated what channel was used to trigger detection. And in most cases, people just use forward scatter because that's what people are used to using for analysis of cells. The flow rate of the instrument for volumetric quantification is important. Issues here are really identical to those for cell analysis, and there's a number of well-established ways to calibrate flow rate. Those should be followed. There are also approaches to calibrate fluorescence and light scatter intensity. I'm just going to segue into a brief survey of fluorescence calibration approaches. There are commercially available calibration particles that everyone should use in association with their EV flow cytometry measurements. There are three types. There are so-called MESF standards, mean equivalent soluble fluorochromes. These are beads whose brightnesses can be traced to an external standard of fluorophore. For instance, fluorophore standards provided by NIST. These are commercially available, and they allow one to calibrate the arbitrary fluorescence intensity axis that's reported by the instrument into absolute units of molecules per particle, molecules of fluorophore per particle. It's a simple linear regression transformation that's made easier by some of the third-party software packages. So in the ideal case, you run your beads at your calibration beads at the same conditions as you did your unknown measurements, and you can convert that arbitrary fluorescence axis into units of molecules per particle. And this greatly increases the value of the data you report to the reader and other researchers. MESF beads are available for several fluorochromes, but not for all. There are multi-fluor, multi-intensity beads, so-called rainbow beads, that are relatively inexpensive, quite stable, and fluoresce on all channels. These are useful, part of the calibration toolbox. The caveat to remember with these is that they are not dyed with the actual dyes that you're going to use in your immunofluorescence experiment. Spectra down here shows the spectra of FITC and PE, and then the spectra of a rainbow type particle, and you see that while it emits in both the green and the yellow, it doesn't have the same spectra as the green and yellow of FITC and PE. So the calibrations performed with rainbow beads will be instrument specific. But a practical approach is to cross-calibrate your rainbow beads against MESF beads on your instrument. And you will have a set of stable calibrated beads for standardizing your measurements. A third type of bead that's useful and also commercially available are antibody capture beads. These are beads that will capture whatever antibody you incubate them with onto beads which have calibrated capacities. So then once you've stained these beads, you use them just like you would use the other standard beads. The advantage here is that you're actually using the antibody that you use in experiment, and so if you have antibody conjugate for which MESF beads are not available, which is the case for many popular immunofluorescence labels, you can use antibody capture beads to calibrate your measurements. So this concludes our brief overview of fluorescence calibration beads. This is really important. Report calibrated measurements. Many people use core facilities to access flow cytometers, and well-operated core facilities will provide these calibration beads and calibrated instruments for users. If your core facility does not, you should go ask them. You're paying for it, and it's their job. So you should tell them you would like to run your samples on a calibrated instrument, and see what they say. There's fluorescence intensity calibration, which is a very important part of proper flow cytometry instrument operation. A few words on light scatter. So light scatter is more complicated than fluorescence. And the reasons for that are that light scatter intensity depends not only on the size of the particle, but also on the wavelength at which you're interrogating it, the angle at which you're collecting the light, and perhaps most importantly, the refractive index of that particle. And so if one surveys the literature, one will find cases where people used beads to set gates for identifying vesicles and used scatter signals from different size beads to try to say something about the size of the particle, the vesicles they were measuring. It's increasingly appreciated, but not universally appreciated, that approach isn't valid. And because of the differences in refractive index between beads and vesicles, such estimates are not accurate. Polystyrene bead might scatter 10 to 100 times more light than a similarly sized vesicle, depending on the measurement conditions. However, so scatter is complicated, but it's also reasonably well understood, and it's possible to use the principles of light scatter to develop models that predict what the scatter would be expected from a low refractive index particle like a vesicle. And there are software available to do this. This is a screenshot example from one of these. This is Josh Welsh's CMPass software. Edwin van der Poel in Amsterdam has also developed a software system to analyze this. So it's still complicated, but there are tools available to make it more accessible. And I think the forward-looking vesicle analysis lab will be incorporating these approaches. Okay, so that had to do with calibrating the instrument. We've prepared our sample. We've stained it with all the appropriate controls. We got our instrument set up and calibrated, and we've made some measurements. And now we want to interpret those measurements. And so from our light scatter or fluorescence measurements, we might want to estimate the diameter, size of our particles. There's a number of different ways to do that. I just described some of the tools that are available for using light scatter. We would consider sort of a advanced area of flow cytometry. If you are going to use those measurements to estimate size, you need to describe in detail how you did that. So good description of the assumptions and models that went into your size estimates is required. Similarly, we can foresee a day where flow cytometry measurements will report on refractive indices of different particles, and likewise, it's going to be necessary to describe in sufficient detail how you arrived at those estimates. Epitope number is an estimate that can be derived from immunofluorescence. If you know how many antibodies were bound per particle, You may be able to estimate how many antigens were actually present. It will involve some consideration of things like, is your antibody, bivalent? Does it bind one or two antigens at a time, or is it a monovalent antibody? So these are topics that, one could discuss for hours, and we have and will again in the future. It's important to recognize the importance of reporting and make these estimates. Finally, we've interpreted our data. We're going to report publication. There is a MIFlowCyt checklist, a general checklist for how to report on flow cytometry measurements. These are additional elements specific to vesicle analysis that should be reported. It's important to not only report the measurements you made, but give the reader some idea about what the sensitivity and dynamic range of the assay you used was. Important to report your EV numbers in terms of a number or a concentration, notwithstanding the pervasive habit in flow cytometry of reporting things as percents. Percent positive, typically the way people will report immunofluorescence data of cells. That's a condition where you're reasonably sure what the denominator is. You know how many cells you detected, so a percent actually has meaning. In the case of measuring small particles like vesicles, in most cases, we know we're not detecting all of the vesicles. We're missing some of them, the smaller ones most likely. And so the denominator is not well defined, and a percentage thus doesn't have much value. So it's really important to actually report the concentration of particles that you measured. And likewise with fluorescence. We can report fluorescence as in the arbitrary units that the instrument reports, but that's of limited value to almost any reader. But if you do calibration and report that brightness in units of MESF or molecules per particle, it ends up having a lot more meaning to the reader and to someone wanting to reproduce your results. And finally, you should share your data. Many fields require depositing data in a public repository. The field of flow cytometry has the flow repository. The working group recommends that you share data associated with a publication via in a public repository. So that's a quick run through of the guidelines. I mentioned each of these categories and the individual topics within them have all been designed or arrived at address a specific artifact or challenge or problem in measuring EV of vesicle, measuring vesicles by flow cytometry. And being knowledgeable about the guidelines and adhering to them going to increase the value of the work you publish. So this is a final summary of the key sort of elements that should be reported. And the most appropriate place to report these is in supporting info for a paper. And it's been suggested that we do this in tabular form, where as a checklist, you make some notation about what steps you took to address each of these components. So the hope is that having researchers on the same page as far as what are the critical elements to report, will increase the value of the published work, but this will also have value, I think, as an educational tool for new people entering the field. If a person doing flow cytometry of EVs for the first time is familiar with these guidelines, they may learn something they didn't know. Flow cytometry is a fairly complicated business. There's lots of ways for things to go wrong. There's no reason for everyone to discover these things independently. So with that, I'll end. And we'll be happy to take some questions that will hopefully start some discussions that will last into the future.
Thank you very much, John. Yep, thank you. Wonderful presentation on the MIFlowCyt-EV framework. We do have a lot of questions coming through, so I'll start by with a question about the detection sensitivity of flow cytometers for EVs. How is it influenced by the laser power from a light scatter standpoint?
So, all things being equal, more light scatter equals more signal. So for light scatter, you can turn up the laser as much as you want, and you will get more signal. That is, light scatter will increase in proportion to the laser power. Unfortunately, so will your background. And so establishing the optimal laser power for a scatter measurement needs also to include consideration of background. Fluorescence does not increase indefinitely with laser power. And so you will eventually saturate dyes, and you may actually even begin to photobleach them. So in some cases, more laser power actually results in less fluorescence. And since we're often measuring both light scatter and fluorescence off the same laser, it's not as simple as just turning up the laser.
Okay, thank you. The next question was with regard, there's actually two questions here about detergent treatment. One is whether or not you have a specific recommendation, and the other is with regard to differences seen between lysing from purified EVs versus EVs in direct labeling of serum containing.
Yep. Good points, both of those. So there's a few papers out there on different detergents. In my lab, we've used Triton X100 and more recently Empigen. Empigen's a little easier to pipette and appears to be as effective as Triton. The efficacy of detergent in lysing vesicles, is concentration dependent, and so for any assay, any system, a detergent titration is a prudent preliminary step. Different vesicles do have different sensitivities to detergent. Interesting feature you will discover if you do those titrations. Purified vesicles, yes, will behave differently with detergent, whether they're in the presence, whether they're in just buffer or in a complex fluid like plasma or serum. Probably the biggest effect there is that there are things in plasma and serum that can also bind to detergent. And so simple way to think about it is you just need to add more detergent to get an effective concentration necessary to lyse vesicles. I guess another just thing to note is that the vesicle the detergent treatment is it's not a perfect control. So depending on the approach you're taking to measure your vesicles, you will find that if you do the reagent-only control that we discussed, that just adding detergent to a buffer sample or a reagent sample will increase background. We've observed. So we use detergent treatment in sort of a semi-quantitative way. We'd just like to know that 90% of the particles we measured were detergent labile. Often there's a ambiguous set of events that can be hard to interpret. If you're really interested in those detergent-resistant particles and understanding whether there's some special type of vesicle or not, will take more work, and things can get complicated. But at the simplest level, just being able to show that most of what you detected is sensitive detergent should increase confidence in your interpretation that what you were measuring were actually vesicles.
So there's a couple of questions here about antibody concentration, perfect way to stain EVs, how many EVs, what final antibody concentration, what volume, how long, what temperature. So there's a number of questions around antibody staining.
Yeah, and so I guess I'll just make the comment that most of the rules that you learn from flow cytometry of cells will apply here. You do need to titrate your antibodies. It's maybe even more important for EV measurements, where we often try to avoid a wash step. In flow cytometry of cells, it's trivial to wash away free antibody by low-speed centrifugation. It's not trivial to wash away free antibody from a suspension of EVs. And so many assays actually just use dilution rather than physical separation. But that means you'll have some free antibody left in your sample when you're measuring it, and it's important to know that that's low enough not to provide an undesired background. So having that antibody titrated well. A good antibody is required, a good monoclonal. There might be 40 or 50 different fluorescent conjugates that you can get used for flow cytometry of cells. In practice, only a few of those are bright enough to do a good job of measuring in flow, of vesicles in flow. So, bright dyes like phycoerythrin, PE size 7, some of the brilliant dyes are widely available, which is nice. Those are the ones you'll want to start with for flow. As you build out larger colored panels, it's important to consider what the effect of fluorochrome brightness will have on the sensitivity. And that's where reagents like antibody capture beads can come in handy to assess what sort of sensitivity compromises you're making by moving to a less bright fluorochrome. So there's I think easily going to be another webinar full of antibody recommendations and observations. That's my short answer.
It's never a short answer. Okay, so there's a question here about the cutoff value for EV size for specificity.
So I'm not sure what that means. Joanne, can you interpret that for me, perhaps?
I'm not sure myself, but I think maybe what they mean is how small or how large something when it's no longer considered an extracellular vesicle.
Okay. So vesicles are metastable structures, formed from phospholipids There is a lower size limit below which it's physically difficult to form a stable vesicle. Reasonably well understood from the biophysics literature, and it's probably about 30 or 35 nanometers. Below that size, it's just the radius of curvature is too tight really to form stable vesicles under most conditions. There may be some examples for that, but some exceptions to that. But in general, I think we can think about 30 nanometers as a lower limit. Of course, there's lots of other particles that you'll find in plasma or other biofluids that are as small as that, including lipoproteins and other interesting nanoparticles. But in terms of vesicles, 30 nanometers is probably a lower limit. On the higher end, it's sort of a philosophical question about what constitutes an extracellular vesicle. There are examples of cells releasing extracellular vesicles that are microns in size. And so size distribution in principle overlaps with that.
So the person that asked that question's just corrected what's been said and said, what is the smallest size of EV that could be detected by nano flow cytometry?
So that will depend on the assay and the instrument. And I would say that's an interesting area of discussion. It will depend on the assay and the instrument. It will depend on the sample as well, various sources of background. There are reports of being able to measure vesicles as small as, I guess, 30 nanometers in really clean, well-controlled systems. Depends on whether you're using fluorescence or light scatter as well. I would say this is a really interesting question, and one that doesn't lend itself to an easy answer. So we'll I think that should wait for a future webinar.
Okay, I have a devil's advocate question here. Why can't I simply use submicron standards for size gating?
The reason you can't use a polystyrene bead to set your gates is because whatever gates you set will be instrument specific, because the light scatter response of different instruments will be different. So on your instrument in your lab, using a polystyrene bead as a reference point might be valid. The scatter from that bead won't reflect the scatter to be expected from a similarly sized vesicle because of differences in the refractive index. But for purposes of setting up your instrument the same way every time, your one instrument, that approach might not be invalid. But if you wanted to transfer that information to a different instrument, that would no longer be the case, and you would need to consider the wavelength, the angle of collection, the refractive index, and all the other elements that affect light scatter intensity.
Okay, we're on to some questions about MESF calibration. Someone's asking, I'm analyzing EVs for the first time. Do I have to do the calibration with MESF for every fluorochrome?
If you think that what you're measuring using that fluorochrome is an important feature of the particle, then yeah, you should report it in calibrated units. So if you're using one of the popular bright fluorochrome, FITSI, PE, FITSI maybe not meeting the criteria of bright fluorochromes. But you can get MESF standards for the most popular, some of the most popular immunofluorescence labels, but many you cannot. And for colors where there is no MESF standard, you have a couple of options. One is to use the EARTH standard or equivalent reference fluorochrome. That's one possible way. The other way is to use the antibody capture beads. So the beads are calibrated, you stain with the antibody you're using, and you can use that to calibrate any fluorescence signal. If you're not doing immunofluorescence, if you're doing some other sort of staining that can't use antibody capture stain, at least reporting the reference brightness from rainbow type beads allows someone to have a chance at reproducing your work, even if we're not able to reduce it to those absolute units of molecules per particle. And so, yes, you should calibrate. You should do the best job you can, because it just adds greatly to the value of your data.
So along those, go ahead,
We'll
Josh.
just have one more question. I just want to remind everyone, because there are a lot of questions that are being asked on the WebEx platform. All of these are being transferred over to Slack, and someone will answer all of your questions, either John or someone else from the working group, or even others listening to this call. So please do join the Slack channel to see any responses to questions that haven't been answered by John.
Okay. There were some questions about the antibody capture beads with regard to clone differences, isotypes, FP ratios, and so on, in accuracy of reporting antibody binding capacity.
Yep. So the antibody capture beads, depending on what type you're using, have some sort of antibody capture molecule on the surface. It's true that all antibody clones don't behave identically. In our experience, most do, but occasionally you'll find a clone that for one reason or another doesn't bind to the capture molecule as well. So that is a limitation of the antibody capture bead approach, is that you could have some variation. In our experience, it's not a large source of variation. It's not anywhere near the largest sort of variation in a typical measurement, but it's there, and you should be aware of it when you use this. A nice thing about the antibody capture beads is it accounts for things like the F to P ratio of the antibody, or the relative quantum yield, which is another important metric of an antibody. Sometimes the fluorescence of fluorophore changes when it's conjugated to an antibody. When you use the antibody capture bead, all of those things are accounted for, and you directly calculate antibodies bound per particle. If you're using MESF values and reporting brightnesses in MESF, then you need to consider how many Fs were on each antibody and work that into your accounting for how many antibodies were bound. This is not too complicated, and it's all been well established over several decades of cell measurements. And most of those lessons apply here to vesicles. I guess the only comment I'd make is that most of the commercial reagents were designed to measure cells, not vesicles. And so the MESF standards and the antibody capture standards tend to be brighter. And so one often ends up extrapolating down into lower ranges of intensity. The linearity of fluorescence response is not the worst assumption we make in flow cytometry. And so while it would be nice to have standards that bracket your brightness, it's not always possible. I'll just note that there are some reagents becoming available commercially that are better suited for the intensity range of vesicles.
Well, that's wonderful. I think we're just about out of time. Thank you so much, John, for presentation and for answering questions. There are a number of questions, and so I hope that the attendee jump into the discussions. Some people may be able to answer questions or continue the discussions of some of the questions that are